My name is Dr. Paul Morris, and I'm the director at Bowling Green of the Kids Tech University program. And together with Jane Roth and Kathy Boer of 4-H programs for Wood County and Ottawa County, we've put together this program for this year. And I'm really excited today to have a morning and an afternoon to have you learn more about fossils. So, how many people here have a fossil? Oh my heavens. So, I know we're going to have a great crowd here. When I was about your age, my father was a farmer, and my first association with geology was every spring he would send us out to the fields to pick up rocks. Little rocks, medium-sized rocks, some kind of big ones. And the first thing I learned about geology was when we picked up the rocks one year, he'd send us back to the same field the next year, and there were more rocks. I knew we weren't growing rocks. I was pretty sure we hadn't missed them because some of them were too big, but they just kept appearing. So that was the first geological question that I pondered. And like a lot of you, I became interested in geology, mostly perhaps because I'm a biologist, fossils, but other things. And I started to read books about geology, and as it happens, some of the books that I read were by a professor by the name of Stephen Jay Gould at Harvard. And this morning we have someone who actually worked with Stephen Jay Gould, Peg Yacobucci. Now, what I can tell you about Peg is she said she's always been interested in fossils. And when she went to university, she went to the University of Chicago, and from the University of Chicago, where she did an undergraduate degree, she went to Harvard to work with Stephen Jay Gould. Now, the thing that I liked about Stephen Jay Gould was that he was a great storyteller. I learned a lot about some really interesting periods in history because he had this ability to weave a story together and make it really interesting. And this morning, you're going to hear from Dr. Yacobucci, an associate professor in our geology department here, and I think she has that unique skill. So I'm going to turn the platform over to Peg because she has lots of interesting stories to tell us. Wrong button.
Just pop it in there. There we go. Ha ha, now you can hear me. Hi. How y'all doing this morning? Really, you're pretty punchy. Okay. So my understanding is you've all been here to Bowling Green before. Is that true? You've all been here before? Okay. Now, let's see. My understanding is that you've heard a couple of these talks before. Is that right? What were they about? What did you talk about before? What did you talk about before? Cells. You heard about cells. Okay, that's interesting. What else did you hear about? What did you hear about? Why are tomatoes red? Why are tomatoes red? Genes. Because of genes. Okay, that works. Why else? Way in the back. What else did you hear about? Fractals. You heard about fractals. Oh, interesting. Okay. What else have you heard about? Fibonacci numbers. That might come up today. How about that? Okay. What else? Yes.
Then, after we heard that talk, then that show Touch came out.
Oh.
And that had to do with that.
Okay. So the TV show Touch. Have you watching that show? I'm not watching that show, but I've heard about it, and it's got numbers and stuff like that, patterns of numbers. Interesting. What else have you learned?
The game of life.
The game of life. What's that about?
It's basically, there are these squares called cells, and you click on them and turn them yellow, and those ones are live, and one that has one or no live neighbors dies or stays dead. One with two live neighbors stay in the same state that it already is in. Three will make it alive, and four or more will kill it.
Okay, so it's a game, and you've got a grid maybe with, and then an individual cell, and depending on what's around it, things can happen. So there's rules to this game involving the cells. Interesting. Okay, well, hang on. So what we're going to talk about today is kind of a little bit different, but also related in a way. So first of all, I'm Peg. Hi. You can just call me Dr. Peg. Dr. Yacobucci takes way too long to say, as you could imagine. So you can call me Dr. Peg. I'm a paleontologist. More particularly, I work on fossils from the Cretaceous period. We're going to talk about the Cretaceous today. Now, I want to say a little bit about my field. First of all, why my field is the best field ever and better than any other kind of science that you've heard about so far. So you've heard these other talks, but mine's going to be the best, just so you know. And the reason it's going to be the best is because, why they saved the best for last, my field is all the other fields rolled into one. So, as an example, how many of you are interested in biology? You like biology, right? Raise your hands, you like biology. How many of you think maybe you could be a biologist? You're thinking about being a biologist. Some of the hands went down, but that's okay. How many of you are really interested in physics? Physics or astronomy? Oh my gosh, really interested, double interested up here in the front in physics and astronomy. Where are the girls? I want more girls interested in physics and astronomy. Thank you very much. What about chemistry? Wow, holy cow, a lot of you like chemistry. Okay, thinking about maybe being a chemist, maybe as a career, something you might major in or be a chemist. Okay, what about engineering? Engineering too, okay, so sort of applied areas of science. Math, computer science. Yes, computers. You like everything, my goodness. Computer science, okay, fair enough. Geology. Okay, here's the thing. If you're a geologist, you are a chemist, and you are a physicist, and you are a biologist, and you are an engineer, and you are a mathematician, and you are a computer scientist. You get to do all of those sciences when you're a geologist. Now, I'm a paleontologist, and I work with fossils. Fossils are our evidence of ancient life as recorded in the rock record. So I'm a geologist. I study rocks. I study the geological record of our Earth. But I'm also a biologist, right? Because I study life. It's ancient life, it's extinct life, but it's life. And I ask biological questions about things. So I'm a geologist, I'm a biologist. Now, the kinds of questions I ask are answered using principles from geology and biology and chemistry and physics and engineering and math and computer science. I do all of those sciences in what I do in my research. I even do a little bit, I dabble in genetics even. So I do everything. That's why geoscience and paleontology in particular, in my view, is the best science of all. Because you get to do all the sciences if you're a paleontologist. So the way I like to think of it is the geosciences, the earth sciences, are the best science if you're interested in big picture questions. If you're concerned with understanding really big scale questions, like that one. That's a pretty big scale question. What can fossils tell us about our planet's past, present, and future? That's a pretty broad question. There's a lot of different things we could do to answer that question. If that's the kind of stuff you're interested in, then paleontology and geology are good fields to go into. So what I want to do is tell you about some of the big picture questions that I'm interested in as a paleontologist. And to do that, well, first I got to tell you a little bit about fossils. So we're going to talk a little bit about fossils in general. What are they? I'll show you some cool pictures. And then I want to talk about some of the questions that I'm interested in my own research that are also questions that a lot of other paleontologists and geoscientists are interested in. And then we'll end with what the future of paleontology might look like. So first of all, what are fossils? The nature of fossils. This is probably the most text-heavy slide you'll see this whole talk. But what are fossils? Fossils are any evidence of ancient life that's recorded in the rock record. It can be anything. So any kind of trace that an organism leaves in rocks counts as a fossil. We might, for instance, talk about body fossils. Body fossils like this dinosaur. People here like dinosaurs? A little bit? Kind of like dinosaurs? Yell if you like dinosaurs. I kind of figured you might like dinosaurs. So body fossils are fossils of, well, what it sounds like, the body of a dead organism. That's the one thing you got to think about when you're a paleontologist. You're mostly looking at dead bodies of things. So you can't be too squeamish. On the other hand, mostly they're really, really, really dead, like this one, which is good for me, because I am actually really squeamish. The first time I ever had to dissect something, I was in 5th grade, and I had to dissect something, passed out cold. Woke up in the hallway, the teacher sitting over me, like, oh my God, what just happened? I'm really squeamish when it comes to very recent dead bodies of things. I like them like this. None of the squishy bits left. That's much better. So most fossils are great for me because they don't have the squishy bits left. It's stuff like bones, teeth, shells. The hard parts of organisms are what's usually preserved. Plants, plant tissues, wood, and other plant tissues might be preserved. Things like insect carapaces, the crunchy part of a cockroach when you step on it, might get preserved. And soft tissues are rarely preserved. There's another kind of fossil, though, that you might not think about right away. And these are fossils that form from organisms that are still alive when they form. And these reveal the behavior of living organisms from the past. And we call those trace fossils. So they're a little different than body fossils. An organism was alive in the past and did something that left evidence of itself in the sedimentary record, in the rock record. So like footprints, these dinosaur footprints here, were left by a dinosaur that walked along a muddy shoreline long ago and left its footprints in the mud, stomp, 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 and kept going. So the dinosaur didn't die then. Its foot isn't there, but its footprint is. Trackways of dinosaurs, burrows of various animals, eggs and nests, and what are some people's favorite trace fossil, coprolites, fossilized feces, dinosaur poop, things like that, which does fossilize on occasion. There's also regurgitites What's a regurgitite, do you think? What do you think a regurgitite is? Yep, exactly. It's what an organism throws up, and then that pile of debris gets fossilized. And we do have fossilized regurgitites on occasion as well. Sometimes we do get soft tissue preserved. Not often, but sometimes. These are real fossils. These are insects. This is a fly and this is an ant that have been preserved in amber. Amber is fossilized tree sap, where a tree is oozing sap, usually a pine tree, some kind of relative of a pine tree. It's oozing sap, and the sap covers up a small animal, like an insect. A bit of plant might get covered up. And then the sap drops to the ground and gets buried, gets fossilized. When the sap hardens, it's an amazing thing, tree sap. It basically acts like plastic, and it preserves whatever is trapped in the sap really, really well. How old do you think those insects are? Oh, who am I going to pick on? Let's pick on you. How old do you think they are? Thousands of years old. Older than that, in fact. Let's see. How old do you think they are? Yep. Millions of years old. In fact, those are on the order of 5 million years old, those bugs. They're very, very old, and they're preserved in beautiful detail because of the amber. Now, I have a favorite fossil in amber. It's this one. It's a gecko. 15 to 20 million years old, from the Dominican Republic in the Caribbean. You can still see its little white belly. Oh, poor little guy. Got trapped in sap and preserved. Now, there's other ways to fossilize soft tissue. This is a squid. 160 million year old squid. This one's preserved in a different way, and the soft tissue's gone. Kind of an interesting way of preserving a soft-bodied animal like a squid. You can see the body of the squid, the head, these little bumps right here and right here where the eyes were, and then here are the tentacles. This fossil was preserved when the squid died, settled to the bottom of the ocean, and within an hour or two, probably after it died, a coating of pyrite, or fool's gold, it's an iron sulfide mineral, coated the tissues of the squid. And then the squid decayed away. The soft tissue isn't there anymore. The squid's gone, decayed, like most soft tissue does. But the coating of pyrite remained. And so we're seeing what that squid looked like because of that coating of mineral that laid over the soft tissue of the dead body. This happens sometimes. Usually happens in association with bacteria. The bacteria that are starting to decay away the tissues cause a microchemical environment right around the area where the tissue is decaying that promotes the precipitation of this mineral. And that's how you get this kind of preservation. So the act of decay itself, under the right chemical conditions, can produce this kind of preservation. Anybody recognize this? Anybody ever seen this thing before? Yeah, what is this? It's a baby woolly mammoth. This was frozen up in Siberia, and this does actually happen sometimes. We find frozen ice age mammals up in the high Arctic, in Siberia, in Alaska. This particular one is 37,000 years old. It's got a name, Lubia, is what they named it, Siberian name. There's a National Geographic special about this little guy. It's a female, little girl, baby mammoth. They're kind of shooting her at this angle from the back because the front of her head decayed away. So you don't really want to see her from the front because that whole front of her head's gone. She died, was buried soon after she died, and the place where she was buried has remained frozen ever since. And so she's preserved more or less intact, a little freezer burned, ever since. Now, you can't get really old stuff like a dinosaur, preserved in this way because you'd have to keep it frozen. The moment it starts to thaw out, it decays. And that's what happened to the front of her head. It started to thaw out and decayed away before anyone could find her. Another way to get soft tissue preservation is if the soft tissue is turned into carbon, pure carbon. So here's a marine reptile called an ichthyosaur, and you can see the skeleton. Here's the eye, big eye on this guy, spinal column, fins, and then on the back you see a fin kind of like a dolphin or a shark, and then on the tail you see a tail fluke like a shark. There's no bone in there. You're seeing the shape of the body. The tissue of the body was preserved as a soft tissue film of carbon. The material is altered to basically a dark film of carbon in the rock. These tufts down here in this picture are tufts of feathers from a dinosaur. Because some dinosaurs, in fact, had little tufts or filaments of feather that are preserved, in this case, along the back. This is part of the spinal cord right here, and then the feathers coming off the back. So you can get soft tissue preservation in that way. More often, though, we just have bone or shell or wood, the hard parts of an organism, and the soft tissue decays away. This, though, is a really nice piece of dinosaur bone. So this is a cross-section through a piece of bone. We've sawed the bone in half, and it's from Alberta. It's about 80 million years old. This is a millimeter scale bar, so each one of these little tick marks is a millimeter in size. Very typical look to the bone. The outer part is pretty dense, and then it gets more open and porous as you go into the inside. If you cut through your leg bone would look kind of like that. What's neat about this one is that the original mineral making up the bone, calcium phosphate, is still there, and then the open, empty pore spaces, well, they're still empty. There's nothing in them. A little bit of sediment, that little bit of white there, and there, that's sediment that filled in some of the open spaces. But for the most part, this is essentially unaltered bone that got buried about 80 million years ago. That's a really nice piece of fossil bone. More typically, though, bone looks like this. This is another dinosaur bone. This one's about 180 million years old. And it looks kind of like the other one. You've got a dense bone up here and then all this open space, except the open space has been filled in, this lighter gray, it's been filled in with more mineral. And that's more typical of bone. And you can usually tell when a bone has been fossilized. Someone hands you a bone and says, is this a fossil bone or a recent bone? You can tell because the fossil bone is heavy. You hold it in your hand, you're like, wow, that's heavy, that's substantial. Modern bone, recently deceased bone, is light because that empty space is still empty. And fossil bone, all the empty space is usually filled in with minerals to make it heavy. And that's called permineralization or petrification. So if you hear about petrified bone or petrified wood, that's how it's preserved. This is what bone looks like. Fossil wood looks like this too. The original material of the wood is still there, but all the empty spaces are filled in with more material. There's another way to fossilize a skeleton. This one's really neat. This is a marine reptile from the Cretaceous, called about 100 million years old. So you have a skeleton here. Here's the backbone, long neck and head, paddle fins on either side. And the bone, which is originally calcium phosphate, has dissolved away. It's gone. But as it was dissolving away, it was getting replaced with a different mineral called silica. Now, silica is a really flexible mineral. It makes a lot of things. It makes quartz. It makes flint, like a flint arrowhead is made of silica. But it also makes the gemstone opal. You don't have opal, like an opal necklace or opal earrings. I've never heard of opal. It's a really pretty gemstone, sort of light-colored, kind of shimmery gemstone. Well, this entire skeleton has been opalized. All of the bone dissolved away and was replaced with opal. So this entire skeleton is now a gem. Really kind of cool. Most opals are actually from Australia. If you like gems, go to Australia. Recognize these? Anybody have these? These particular fossils? Where'd you get them? Where'd you find them? Fossil Park in Sylvania. How many of you have been to Fossil Park in Sylvania? There's lots of very nice fossils up in Sylvania at the Fossil Park, and indeed, that's these guys. You'll find pockets full of these fossils up in Sylvania. These are Devonian in age, puts them about 380, 390 million years old, much older than dinosaurs. These are trilobites called phacops. They have big eyes. They're related to horseshoe crabs and shrimp and lobsters. And these are brachiopods. They look kind of like a clam. They're not a clam. They're a completely different kind of animal, but they also have two shells that hinge together like a clam. They're ridiculously common. You find them all over the place up in Sylvania. These are also fossilized, and they're fossilized a lot like that opalized marine reptile was. The original shell material was dissolved away and then replaced with more mineral. In this case, the same mineral the shell was made out of, calcite. But I wanted to show you these so that you would know, if you ever do go to the fossil park, this is what you should be looking for. These brachiopods and these lovely trilobites. You often find just the head of them or just the tail of them, the trilobut of the trilobite. Another way to fossilize things is to find really not the body at all, but an impression of the body. So here, for instance, this is dinosaur skin. Now the skin isn't there, but when the dinosaur died, lying there in the sediment with its skin, touching the sediment, it left an impression in the mud of what its skin looked like. And then, it decayed away. It's gone. The dinosaur itself isn't there. The body decayed away, but the impression of the skin was preserved in the sediment. So we know that while some dinosaurs had feathers, other dinosaurs had scaly skin. And we can tell exactly what the scales were like and the patterning of the scales because we can see it in the sediment. Footprints are often preserved in this way, where the animal stepped down into the sediment and then went on about its merry way. And we have a mold of what the foot looked like without ever actually having the body of the animal at all. We just have an impression in the sediment of the fossils. Now, I've been talking about dinosaurs, of course, and some brachiopods and some trilobites, but people forget sometimes that there are other things in the world. In particular, there are microorganisms, bacteria, single-celled plankton, and they have fossils too. In fact, the earliest evidence we have for life on Earth as fossils are these things. These are stromatolites. These are layers of photosynthesizing bacteria that make basically a film or a mucousy scum on the sediment in shallow water, and the sediment binds to that sticky scum, and it layers over time to build up into these dome shapes. The oldest evidence we have of life on Earth comes in the form of things like this, stromatolites. It's how we know that there were photosynthesizing bacteria as long ago as 3.5 billion years ago. This is a fossil of a single bit of bacteria. You take a rock, you slice it very, very thin, you put it under a microscope, and you can see cells, fossilized cells. Now again, the actual soft cell is gone, but the outline of the cell is preserved by mineral coating it. Over here, these are shells. These are the skeletons, the hard parts of single-celled plankton. These are diatoms. These photosynthesize, and they're found in all sorts of different aquatic habitats, freshwater and ocean habitats. They're absolutely gorgeous. They make all sorts of different forms of silica. They use silica to make their skeletons. Beautiful, beautiful shapes. Again, looking under a microscope to see these. And these are forams, or foraminifera. No one says foraminifera, everyone just calls them forams. These are made of calcium carbonate, the same mineral that's used to make brachiopods or trilobites or clams or snails. But these are single-celled plankton that live up in the ocean. They don't photosynthesize, they actually eat those guys. They eat diatoms and other single-celled plankton. Now, while we love dinosaurs, and I will talk more about dinosaurs, I promise, I want to talk about these guys a little bit. Because it turns out, for some of the big picture questions we're most interested in, these guys, the forams, are of special importance. Because one of the things we're most interested in documenting as geoscientists is how our planet has changed over its history. How has the physical condition of our planet changed? How has the environment changed over its history? And one of the best ways to get at that is to look at microfossils. All sorts of different kinds of these microscopic organisms that are preserved as fossils. So for instance, we might, if we're interested in terrestrial, land-going deposits, look at fossil pollen. This is a single grain of pine pollen. If you have allergies, you hate pine pollen. It can make you sneeze. That's a single grain of it. If you sample sediments and look under a microscope and pick out all the little grains of pollen, you can look over time and see how the plant community has changed. When in Ohio would we have lots of spruce trees? When it was much colder. When did we have very few spruce trees and more shrub vegetation? When did we have lots of grasses, lots of sedges, and so on? So we can track climates and habitats by looking at pollen. We can also look at global climate. If we look at these forams, here's another view of forams, a scanning electron microscope view of forams, these little microscopic fossils. And this is a microscope side. I just want to show you how big these are. These are relatively large forams. You can just see them with the naked eye. The smallest ones you really can't see at all. The biggest ones, on a standard microscope slide, you can just kind of make them out. It's about as big as they get. So they're pretty small, but there's billions upon billions of them in the ocean. And so when you're sampling ocean sediment, you see them, and you can sample them and use them to document our climate. The way this works, here's an example. Here's some sediment core from Greenland. So you go off the coast of Greenland, and you sink basically a big tube into the seafloor. And you draw the mud from the seafloor back up. So here's columns of mud, and you just kind of read this, stack it all back up together again, and read it down. And you may be able to make out that there's some layers in here, up through, and some parts are darker, and some parts are lighter. You would go through that sediment core bit by bit, and you would sample, and then sieve that sediment and pluck out guys like this. This is another foram, Neoglobal quadrina pachyderma, if you want the name of it. It's a lovely thing. They're very beautiful to look at. Very aesthetically pleasing little fossils. Which is kind of sad, because the way you study these, mostly, you take these fossils and you grind them up to powder. Kind of sad, really. Grind them up to powder, and then you do chemistry on them. You look at the relative amounts of different isotopes of oxygen in their shells. Because the amount of oxygen, different isotopes of oxygen in their shells, reflects the temperature of the water in which these were growing. And that's incredibly useful. Because if you're interested in how climate is changing over time, you can get these fossils, extract them from these sediments in the ocean over a long geologic time period, crush them up, look at the oxygen isotopes, and then from that, calculate temperature. What was the temperature of the Atlantic off of Greenland over millions of years? And that gives us records like this one. This is a graph showing you the present day on this side 1 million years ago, 2, 3, 4, 5 million years ago. And then the squiggly line that keeps zigzagging back and forth is the record from those forams. Crushing them up, measuring the ratio of oxygen isotopes to get temperature, and then plotting it. And you can see, wow, it changes a lot, first of all. The amount of variation in our climate is really strong, and it really wobbles. It was pretty warm overall until about 2.5 or so million years ago, maybe 2. And then it's colder, times when it gets colder, and then warmer, cold, and warm, cold, and warm, cold, and warm. And then in the last million years, there's been a lot of times when it's been quite cold. But again, seesawing back and forth. Welcome to the Ice Age. The last 2 million years, Earth has been in an ice age with large ice sheets in the northern hemisphere. Before about 2.5 million years ago, there was no ice sheet. There was no big ice cap in Greenland, no substantial ice in the winter, in the summer, in the Arctic Ocean. So that's what the world looked like. This map is showing you what the world looked like before 2.5 million years ago. And then these two maps are showing you those wobbles, times when it was relatively warmer, oh, but still a big ice sheet on Greenland, like today, and times when it was relatively cold and we had a big ice sheet here in North America. That's what the Ice Age is. And it's these foram fossils that allow us to document with great precision exactly how climate is changing across that 2 million year interval. Here's another view. This is down at the North Pole. Here's the modern day view. Here's the North Pole. Here's the Greenland ice sheet. Here's North America. The Great Lakes aren't shown on this, so I'm going to follow the St. Lawrence down, and we're right about here. That's about where Ohio is today. So today, not a big ice sheet, but there is an ice sheet. We're still in an ice age. There's still a big ice sheet here. It's just not as big as it was only 18,000 years ago. We would have been sitting under this ice sheet. Big, 1 to 2 kilometer thick ice sheet sitting on our head here in northern Ohio. That was the extent of it. Covered all of northern North America into Alaska. Another ice sheet covered Europe over into Russia. So very, very recently, climate was radically different than it is today. Of course, that means Ohio was pretty different than it was today. Sadly, you may already know this, you're never going to find a dinosaur in Ohio. There's no rocks of the right age for dinosaurs here in Ohio. But that said, you might find one of these. Ice Age mammals are found in almost every county of Ohio. So you might find a woolly mammoth, or a mastodon, or a giant ground sloth, or a peccary. This is a peccary. You legitimately might find those. Back in my lab, I have bits and pieces of a mastodon that was found 2 miles from here in Bowling Green, as an example. This afternoon, in some of the activities you're going to do, I have some fossil mastodon and mammoth material you're going to be able to look at and pick up and touch. Get a sense of what that stuff looks like, because I want you to know what it looks like, because you might actually find it. And I want you to know what to do with it if you do find it. So this is the kind of stuff that lived here in Ohio as recently as maybe 10,000 years ago. And then they all became extinct. All of these large mammals disappeared. And there's much debate about why that happened. Now, I'm talking about an ice age. You should know that the Earth has not always been this cold. This is another plot. It's showing, again, temperatures based on forams, those fossil microfossils crushed up. Present day is at the top. And now we're going back in millions of years, 10, 20, 30, 40 to 50 million years ago. Cold is over here. We're cold. We're as cold as we've pretty much ever been right now. And then if we go this way, it gets warmer. And you can see it's only been the past couple million years it's been so cold. It was much warmer only, say, 5 million years ago, or 10 or 20. And we go back to 50 million years ago, it was much, much warmer on Earth than it is today. Okay, fair enough. That's 50 million years. I have a question for you. 50 million years is a long time, right? 50 million years is a long time. I want to know how long that really is, though. So relative to the history of the Earth, the entire lifespan of our planet, how old do you think 50 million years is? Is 50 million years half of Earth history? How many people think it's half of Earth history? I'm getting shaking heads now. How many people think 50 million years is maybe a tenth of Earth history? So maybe it's 10% of Earth history. A couple hands go up. How many people think it's less than 10%? Good. In fact, it's about 1% of Earth history. There's Earth history. Show you a time scale for the entire history of our planet, 4.5 billion year history of our planet. Our planet and the rest of our solar system formed about 4.5 billion years ago. There's the present day over there, 0 billion years ago. Here's 4.5 billion years ago. 50, in this time scale, you got to think about this. Here's 0. This would be 500 million years ago here. So 50 million years ago would be a tenth of that, would be right about there.
Wow.
Yeah, so that plot I showed you doesn't go back very far. Now, I wanted to point out a couple of key things in the history of life on this time scale. There's a lot of other stuff on here too. The oldest evidence we have for life on this planet is between 3.5 and 4 billion. So the oldest stromatolites, the oldest evidence of fossils, this guy here is one of the oldest fossil bacterial cells, they go back to around here. So 3.5 to 4 billion years, that's the oldest life. But it's bacteria. And it's bacteria. And so we're going to keep going. 3.5 billion year bacteria, 3 billion years, it's bacteria, and nothing but bacteria. 2.5 billion years, it's bacteria, we're waiting. We have to get way up here between 2 and 2.5 billion years before we get anything on Earth other than bacteria. That's amazing. This planet is a bacterial planet. Bacteria were the first things to appear on this planet. They are still the dominant life form on this planet. We like to think we're the dominant life form on this planet, but really it's bacteria. They're still dominant. They're in more different environments. They do more different kinds of things than any other form of life. They've been here the longest. We get other things, more complicated life, 2 to 2.5 billion years ago, more complicated single-celled organisms, the first plants, but ocean-going plants, not land plants, seaweeds and things, about 2 billion years. And we keep going, no animals. We're still waiting for animals. We don't get animals till here, just a little more than 500 million years. So animals are late in showing up. And you're waiting for dinosaurs, I'm sure. Well, they put dinosaurs on this, conveniently enough. That white bar is the time when dinosaurs were present. They're pretty late. On the other hand, it's a long bar. Dinosaurs are probably the most successful land vertebrates ever. They lived for the longest amount of time compared to other land vertebrates. They beat us by far in terms of how long they were around. So I wanted to show you this to kind of put a perspective on what we're going to be talking about, because I'm going to focus in on just this last bit of when dinosaurs were around. Yeah?
What's the Zach Hill zircon?
Zach Hill zircon. Glad you asked. That is the single oldest thing on our planet. Meaning it's the single oldest mineral crystal that's been dated radiometrically. It dates to 4.4 billion years old. It's the oldest thing we've been able to date. One more graph, and then we'll talk about some other things. This shows the history of carbon dioxide in the atmosphere going back just to the time of animals, the first appearance of animals. And I just wanted to show you that today we're in a very cold time with not a lot of carbon dioxide in the atmosphere, ice house conditions. And there was another time about 300 million years ago that was similar. There was another big ice age, lots of ice sheets on the continents. But on the other two times, in between them and before, it was a greenhouse world. There was lots of carbon dioxide, a greenhouse gas in the atmosphere, and climate was very, very different. The Cretaceous time period right here is the last part of that greenhouse world, and that's the time period that I work in. I want to talk about that, the Cretaceous. As I said, Cretaceous is a greenhouse world. I put some numbers up there. Atmospheric carbon dioxide levels in the atmosphere, 6 to 10 times as much as they are today. That's a lot. Greenhouse gases like CO2 trap heat from the sun at the Earth's surface, and that means surface temperatures are up to 25 degrees Fahrenheit warmer than they are today. This is a climate model. This is a present-day map, well, 1990s map, and it's showing you temperatures. There's a key for you, but basically blues are cold and warm colors, yellows and oranges are warm. So that would be the temperature we see today. This is 21,000 years ago during the last glacial maximum when there was a big ice sheet. You can see the ice sheet here in North America. Now compare that to what the Cretaceous would have been like and how much hotter it would have been in the Cretaceous and how hot, especially down here in South America, here in what will eventually become the Indian Ocean, it will be. Now, there's another thing to notice. You look at the continents here, North America, South America, Africa. Now compare them, wait a minute, what's going on here? Those aren't the same continents. What? Well, that's what the world looked like in the Cretaceous. Plate tectonics. Pangea, supercontinent with all the continents stuck together, was breaking apart in the Cretaceous. South America and Africa are still stuck together. Antarctica and Australia are still stuck together way down here in the southern hemisphere. This is India and Madagascar still stuck together. Here's North America, Greenland, and Europe, more or less still attached up here. And here's Asia and bits and pieces of Asia, kind of all coming together over here. Now this is the early Cretaceous, 120 million years ago. If I go forward to the late Cretaceous, 90 million years ago, things have changed. I'm going to go back and forth a little bit, pseudo-animation here. For instance, watch India and Madagascar. Keep your eyes on India and Madagascar. Zoop. They move up, they start to split apart. I'll go back again. Watch India and Madagascar. Zoop. Right? I love doing this. You can do this all day. Also, watch South America and Africa break apart. There they go. The other thing, I'll do it one more time. Look at sea level. By the late Cretaceous, sea level reaches its highest point ever in the history of the Earth ever. Because it's so warm, in part, there's no ice, and so all the water that today is locked up in ice sheets is in the ocean, and large parts of the continents are underwater. Look at North America. All this light blue is water. So the ocean went from the Gulf of Mexico right through the middle of North America up to the Arctic. Africa is split in half by ocean. Europe is basically a bunch of islands with ocean in there, cutting up behind the Ural Mountains here in Russia as well. Lots of ocean. Ohio would be right here. So we weren't underwater, and in a way that's bad. That's the reason that we don't have dinosaurs. There were dinosaurs here, of course there were. There were dinosaurs all over here. But there was no sediment being accumulated on this piece of continent. And so the rocks that would have fossil dinosaurs in them here in Ohio have eroded away. The good rocks that have tons of dinosaurs in them were deposited right along the shore of the seaway over here in western North America. So if you go to Montana, North Dakota, Wyoming, Alberta, that's where the cool Cretaceous dinosaurs are. Over here, we don't have rocks of the right age. There certainly were dinosaurs here, but they're just not preserved. Why I'm interested in the Cretaceous is because it's a good model for what might happen in our future. We're all worried about global warming, human-caused global warming due to the human input of CO2, carbon dioxide gas, into our atmosphere. And so what paleontologists and geoscientists are interested in is how do ancient organisms respond to episodes of global warming? We want to know how our Earth, our present-day Earth, is going to respond to global warming, how ecosystems will change, how individual species will respond. Well, how did organisms in the past respond? How did ecosystems change? How did habitats shift? We need to find good models for global warming in the past. And the Cretaceous is about as good as it gets. This is not the Cretaceous. This is a picture from NASA of temperature anomalies in the first decade of the 21st century. So it's comparing temperatures from 2000 to 2009, comparing that to the average temperatures in the 1950s, 60s, and 70s. And where it's orange and red, that's where it's warmer today than it was in the past. And the very few spots, there's a little bit of blue right there, and this is an anomaly due to a lack of weather stations down here. Where there's a little bit of blue are places where it's colder than it was in the past. So we want to use the Cretaceous as a model for what a greenhouse world looks like and how do organisms respond to greenhouse conditions. So this is what I do. What I work on is mostly fossil ammonites. Ammonites are related to squids and octopus and nautilus. And I focus on nautilus or ammonites that lived in the Western Interior Seaway. That's the seaway that cut through North America. So here I am collecting in Wyoming, collecting out of rocks, shells that look like that in the rock and like that once they're out of the rock. That's a reconstruction of what one of these guys would have looked like when it was alive. Basically a squid in a shell. That's how I like to describe them. Here's some pictures of fossil ammonites. They are related to the one living cephalopod today that has an external shell, and that's nautilus. Does that look familiar to you? Yes. You saw that, right? Yes. When did you see that? When did you see that? You saw that in the talk about patterns, right? Fibonacci series boiled in there somewhere. This shell is coiling in a way, a logarithmic spiral. I think your speaker in that first talk talked a little bit about that and how there's the Fibonacci pattern is manifest in the shell. Well, ammonites coiled that same way. They have a coiled shell with partitions, evenly spaced partitions in it that control buoyancy. So the animals sat in the outer part of the shell. Now, some of them coil very neatly. Some of them have lovely ornaments. Some don't behave as well. This is one that does not coil in a normal way, coils in a very strange way. They don't all coil as nicely as nautilus does. But they're very, very cool, and they're very fun to work with. They're very common fossils, especially in the Western Interior. The Western Interior is a lovely place to look for fossils because it has a very rich fossil record. The Western Interior in the Cretaceous was full of ammonites. There's some ammonites, there's some more. Swimming around in the shallow sea with mosasaurs. These are extinct marine lizards. They're closely related to the Komodo dragon. Ever heard of the Komodo dragon? Yeah? This is a very big Komodo dragon that's become strictly aquatic. So it lives only in the water. They got to be over 20 feet long. So very large predatory ones. And you can find ammonite shells with perfect punch marks that match the teeth of mosasaurs. So we know mosasaurs were eating ammonite. Is that working? Okay. Technical difficulties. There we go. Okay. So there's a crocodile in the Western Interior Seaway that was big enough to eat T-Rexes. It would come up, it would lie in wait for the T-Rexes when they came down to the shore, and it was big enough to bite them in half. That's how big this crocodile was. And we know it was doing that because there's dinosaur fossil bone with these crocodile tooth marks in them. So we know it was doing that. It's a very interesting place, the Western Interior. That's what it looks like today, though. Kind of rough to do field work in parts of the Western Interior. This is a site we went to in West Texas. Oops, hang on. There we go. Whoa. More technical difficulties. I'll get it back. There we go. This is a site we went to in West Texas. It's halfway between El Paso and Big Bend. Mexico and the Rio Grande is about half a mile, quarter of a mile over that hill. We're in the desert. There's two grad students sitting there. I don't know if you can see them. They're very small. Two grad students sitting there collecting fossils. This is what we're getting out of the rocks. It's a great place to collect fossils, though, because if you think about it, the seaway drained away. And so all the sediment that accumulated in the seaway piled up, right? And now, because it drained away, it's exposed. And so all the fossils that accumulated in those sediments we can sample. And because we're in the desert, no vegetation, very little vegetation, so it's just sitting there. The fossils are just sitting there waiting to get picked up. Kind of rough. Camping. We camped in tents on this site, hit 120 degrees during the day, got down to about 110 at night. Woohoo! It was pretty rough, that particular field season. But spectacular fossils we were pulling out of these rock units. This is my big picture question. This is what my actual research is on. Why do ammonites evolve so fast? Of all the animal groups, and you start thinking about animals, corals and sea anemones and clams and snails and crustaceans and crabs, why do, of all those groups, this group, ammonites, evolve like mad, really, really, really fast? That's the question. So that's what I focus my research on. I'm studying the anatomy of these fossils and trying to understand why they speciate so quickly and in such a profound way. So we're looking at details of their anatomy to try and track how exactly is it that they're evolving, and also relating it to the environmental changes we're seeing in the Western Interior Seaway. That involves some field work, but most of what I do involves sitting in front of a computer and doing a lot of math, a lot of computing to analyze the anatomy of these things mathematically. So much of what I do is very statistical and using computer programs in novel ways to quantify the patterns we're seeing in the evolution of this group. Yeah, but I can tell you're thrilled with ammonites, right? This is what you want me to talk about, I'm sure. Well, it turns out, I do have a confession. I work on ammonites. They have a spectacular fossil record, huge numbers, right? And they're really cool, and I love them. as an undergraduate, I worked on dinosaurs. And my students and I still work on dinosaurs. I worked on these guys, ostrich mimic dinosaurs. They're still my great love, the ornithomimid dinosaurs. But I do, with my students, still work on dinosaurs. So I just wanted to tell you a little bit about two of my students, recent students, and what they've done with me, working on dinosaurs using engineering and physics principles to study how dinosaurs functioned, what their basic biology was. So this is Liz. Liz Ferrer graduated a couple years ago from BG. She's now working on her PhD at California, Berkeley. There she is with a friend she found in the field. Not a rattlesnake. It's a friendly snake. She loves snakes, so she just picked it up. We're like, put that down. That's her dissecting a rhino leg with John Hutchinson at the Royal Veterinary College in London, where she spent a summer between college and grad school of her own volition. John Hutchinson is probably the leading person who studies the biomechanics of dinosaurs. How do very, very large animals function? And he does that by studying very large animals today, which is why they're dissecting a rhinoceros leg. And she's very, very happy about dissecting that rhinoceros leg. She's much less squeamish than I am. What she did for her senior project, Liz got access to scans, computer scans of Majungasaurus skulls. Majungasaurus is a large carnivorous dinosaur from Madagascar. And Larry Whitmer at Ohio University is working on it. It is about the same size as Allosaurus, if you know what Allosaurus is, but about half, a little less than half the size of a T-Rex. It's big, 20 to 23 feet long, including the tail. It's really interesting that it's quite anatomically different than the other large predatory dinosaurs. The big predatory dinosaurs are not all closely related. Different groups of small predatory dinosaurs independently evolved to be big. And that was her research question. That was her big picture question. How much of the anatomy of the skulls and the biting of big predatory dinosaurs is similar, because they're all doing the same thing, big animals trying to eat things, and how much of it is different because they all evolved independently from different groups? So she did a finite element analysis study, and basically it's an engineering technique looking at the forces that are distributed through the skull of Majungasaurus when it bites down, and found that the way it distributes forces through its skull when it grabs at something are completely different than the way Allosaurus works or a T-Rex works. So that all three of these large predatory dinosaurs are biting prey in different ways. They're attacking prey, they're eating prey in different ways, which is a really interesting question, really interesting problem, because we think we understand how T-Rex and Allosaurus feed, but this pattern was weird, and we still don't quite know what Majungasaurus was doing with its head. How was it actually biting at things? Was it slashing at them and backing away, which is kind of what Allosaurus does, or was it crushing and holding on, which is what T-Rex did. This is Leah King, who also graduated a couple years ago. She also did a finite element analysis study. This is her bonding with a dinosaur fossil. She volunteered, she and Liz both volunteered at a dig site out in Montana run by Jack Horner. You may have heard of Jack Horner, paleontologist at Montana State, and so she's very happily digging up dinosaurs. She worked on something called Thessalosaurus, which is a much smaller, 8 to 13 foot long, plant-eating dinosaur. And her question, you can kind of see it in the feet, there's a debate about whether they walked around kind of flat-footed or were up on their toes. That was her question. Her big picture question is, how is this thing walking? Is it up on its toes or is it flat-footed? So she analyzed the foot bones of these guys to try and figure out what's the best angle, what minimizes the stresses on the bones, and figured out, yeah, they're up on their toes. Just slightly back a little bit from being up, straight up on their toes. But sadly, there are no dinosaurs with us anymore, except of course for birds. Birds are theropod dinosaurs. They evolved from theropod dinosaurs. They are theropod dinosaurs. You might have a dinosaur for lunch today if you have some chicken nuggets. So where do they go? Where did the dinosaurs go? And the ammonites, which died out at the same time as the dinosaurs. The other reason I like ammonites, they're basically the dinosaurs of the sea, I like to think. There's some. Well, we know what happened to them. If we look at the geological record, right at the end of the Cretaceous time, we see a spike in the rock record in an element called iridium. And iridium is very rare at the surface of the Earth, but it's very common in extraterrestrial materials. And to get a big spike right at the end of the Cretaceous can only mean one thing. To get that much of that element that's only common in extraterrestrial materials all at once in the rock record can only mean that. It can only mean that a very large object from outer space arrived at the surface of the Earth at the very end of the Cretaceous. An asteroid hit the Earth. In fact, we know where it hit because we have the crater, Chicxulub crater, in the Yucatan Peninsula in Mexico. This very careful map of surface elevation shows you the edge of the crater here, this part of Mexico. You can see the edge of the crater. It's one of the largest impact crater features known on the planet, and it dates radiometrically right to the end of the Cretaceous. So we know that an asteroid hit the Earth. Now, what that actually does to kill the dinosaurs, well, it impacts the Earth, generates a huge amount of debris. It vaporizes all the rock in the area it hits, which goes up into the atmosphere. This is right after the impact, 60 seconds after the impact. This is 30 days after the impact. The entire surface of the Earth, the atmosphere is clouded over. It's full of debris. And that's going to block sunlight. That's going to make it colder, and it's going to stop photosynthesis. So part of the argument is that dinosaurs froze to death and they starved to death. And if that's not bad enough, that impact also generated a lot of heat and wildfires, and so they also burnt to death. So a lot of bad things happened to dinosaurs and to ammonites at the end of the Cretaceous to kill them off. But could we bring them back? Jurassic Park. What about woolly mammoths? March 13th, 2012, Russian and South Korean scientists are working together to clone a woolly mammoth using cells taken from a frozen fossil from Siberia. Remember those frozen fossils I showed you? In fact, the Japanese have been working on this for more than a decade, trying to either clone a woolly mammoth from frozen tissue or produce a hybrid by taking sperm cells from a frozen male woolly mammoth and impregnating a female elephant, and seeing if they can get an elephant-mammoth hybrid that's viable. It's also been proposed that we should take some of the living relatives of these extinct Ice Age things, like elephants and camels, lions and cheetahs that used to live in the United States until only 10,000 years ago, and put them back. Let's put cheetahs and lions and elephants back in Kansas. They were here 10,000 years ago. Let's fix it. Let's put them back. It's called rewilding. Is that a good idea? Oh, strong opinions. And then there's the dino chicken, proposed by Jack Horner. The dino chicken. He wrote a book about it, How to Build a Dinosaur. Extinction Doesn't Have to Be Forever. My favorite review of this book, it will easily tap into the vast mad scientist market. Can we genetically modify a chicken to express long dormant genes? As I told you, chickens and all birds are dinosaurs. They are theropod meat-eating dinosaurs. And they still have the genes. They're just not expressed. They have the genes to produce a long tail, teeth, separate fingers with claws, lose the feathers and get bigger. They're just switched off. The genes are there, they just don't express during the development of the embryo. What if you turn them back on? That is a picture, not of a dinosaur, but of a modern chicken with a long tail, teeth, separate fingers with claws. It would be about 3 or 4 feet tall. So they're trying it. Montreal, McGill University, the Canadian government has approved the research. They're hoping by 2020 to make a dino chicken, to get a chicken embryo to express the genes for teeth, claws, tail, 3 or 4 feet tall. Now, you may be asking yourself, do you really want to take a chicken, not the friendliest animal, if you've ever met a chicken, make it 3 feet tall and give it teeth and claws? Well, let me give you some arguments for it. First of all, Jack Horner has argued it would be a beautiful demonstration of evolution. To be able to go out in front of an audience like you, talk about dinosaurs, then go off stage with a leash, bring back in the dino chicken, and say, look, evolution. That's pretty persuasive. But also, many diseases that affect humans are due to genes that are switched off when they shouldn't be. If we can do this to a chicken, maybe we can help people who have diseases where the genes need to be turned on, and they need help getting their genes turned on. So there's medical benefits potentially to this research. It's not just a fun game. So I know you want to ask me questions. I'm going to leave you with some take-home questions for you to think about over lunch, and then you can come back and ask. We can discuss these at the activity tables. You can also talk about these with your parents. What's your favorite extinct organism and why? Would you rather live in an ice age or a greenhouse world? Do you like skiing or surfing? Do you think humans will become extinct one day and why? And do you think we should make a dino chicken or clone a woolly mammoth or not? You all changed your mind. Thank you.
Wow, that was great. That's a big hand for Peg. Okay, we're kind of constrained here because we have one microphone. So I'm going to run up and then I have to run back because if we don't give her the microphone, nobody in the other room can hear. So excuse me while I run. We'll start with you.
Can animals survive in the ice? Like if they get frozen and they can't be frozen, could they, like, if they defrost, could they still be alive?
My teacher, she's been saying that people are thinking about sending people to space and freezing them so they can go a while.
Okay, so the question is, if you freeze an animal, will it survive? This is an interesting question. For the stuff we've been talking about, no. So like in natural conditions, for an animal, I would say no. Not necessarily for microorganisms, and that's going to come up, I believe, at the tables this afternoon. You're going to see an example where that may not be the case. For microorganisms. Now, cryogenically freezing a person, and then with technology, and then thawing them out again, that hasn't been successfully invented yet, but people have been talking about doing it for a long time.
The crater on the peninsula in Mexico, if it had enough power to wipe out the dinosaur, why didn't it break off the peninsula?
Okay, the actual impact did vaporize a very large amount of the rock around the area, so the crater is much bigger than the object itself. The object that hit the planet was probably only about 10 kilometers wide. The crater is over 100 kilometers wide, so that whole area got vaporized by this little rock. So imagine a 10-kilometer wide rock hitting and making a hole 100 kilometers big. So it did vaporize a big chunk of the area. A lot of the things that killed off the dinosaurs, though, was the effects in the atmosphere. So the debris that went up in the atmosphere starving the dinosaurs, the fires that spread across the land and things like that, rather than the actual impact energy itself.
Did dinosaurs ever live on the Earth at the same time as humans?
Okay, did dinosaurs ever live on the Earth at the same time as humans? No. The last dinosaurs, let's count birds, dinosaurs died out 65 million years ago. The earliest fossil humans show up about 4.5 million years ago, and our species, Homo sapiens, appear about 200,000 years ago. So there's about a 60 million year gap between dinosaurs and humans.
If we ever did make this dino chicken, would it be able to evolve into like a dinosaur?
Well, it depends on how you think about it. In a way, it is a dinosaur, because birds are dinosaurs, and you're going to make it look more like a dinosaur. The question then is, and that's been asked, are you going to let them breed with each other, interbreed and produce offspring over generations and generations? They could potentially evolve. Whether you'd want to let them do that or not is a question. Whether you'd ever make enough of them to have a breeding population would then potentially, if you put them in the wild, they could evolve, sure.
If the meteor crashed and killed off all the dinosaurs, how did birds survive and other animals be created?
Excellent question. So if the meteor killed off all the dinosaurs and all these other things, why did anything survive, like birds? There definitely is a selective pattern. Large animals died out and small animals didn't. If you were bigger than about 50 pounds or so, you were much more likely to die out. And the small dinosaurs, some dinosaurs are really small, like bird-sized, but by the end of the Cretaceous, the smallest known dinosaur is about human-sized, and so they all died out. The birds at that time, the mammals at that time, most other reptiles at that time were smaller than about 50 pounds, and they tended to survive. That's probably because small-bodied things were better able to burrow to avoid the fires, better able to hibernate, and sleep through the darkness in the period where there wasn't a lot of food. And so small-bodied animals were more able to survive some of the bad conditions. Big animals kind of had no place to hide. You know, a T-Rex isn't going to burrow, dig a hole and hide. It's kind of hard to hide a T-Rex. So it may be a size selectivity pattern that we're seeing.
How many ways are there to fossilize a creature?
How many ways are there to fossilize something? Well, gosh, if you think about it, all sorts of different ways. We talked about a bunch in the first part of the talk, different ways of preserving the bone, different ways of preserving the soft tissue. You can preserve just the impression of the thing in sediment. One thing I didn't talk about is the idea of a chemical fossil, where you're only preserving some of the chemical traces of an organism. For instance, sponges, an animal, sponge, not the sponge like a sponge, but a sponge the animal. We know that sponges are present by about 600 million years ago because there's a kind of molecule, an organic molecule that only sponges make, and we see that in sediments. We don't have the fossil of a sponge, we have a chemical, this particular organic molecule that only sponges make that's present. So that's another way to preserve organisms, at least to show evidence that they were there. If you think about it, even oil and gas, natural gas, coal, that's evidence that life was there. So there's a lot of different ways that we can see evidence of ancient life. If dinosaurs became extinct, then how did humans get going? Well, dinosaurs are wiped out by the asteroid, and mammals are present. One thing that's a common misconception is that, well, once upon a time there were dinosaurs and then came mammals. In fact, dinosaurs and the first mammals appear at almost the same time. And the whole time you have dinosaurs, you have early primitive groups of mammals present with them, living at the same time as them. They're small, rat-sized, cat-sized sort of mammals. When the dinosaurs die out, the mammals start to diversify. And within about, 10 million years or so after the dinosaurs die out, you've got all the major groups of mammals that we see today, from bats, flying mammals, whales, first whales show up, swimming mammals, primates, our group are present, The hoofed mammals are present. The elephant group is present. So all the major groups are present within about 10 million years after the dinosaurs die out, and then they diversify over that about 60 million year interval, and we eventually get humans. What do I have to study to become a paleontologist? Also a very good question. There's multiple pathways to become a paleontologist. For those of you who are interested, how many of you think, hey, I could be a paleontologist? That's interesting. A couple of you. Okay. You have two ways to go, really. Because paleontology is both geology and biology, you can do it through studying geology or through studying biology. My way, I was a geology major, so I studied geology in college and in grad school, but I also took a lot of biology classes. So I did both geology and biology in school. I also took physics, I took chemistry, I took math, I took statistics. It is the everything field, so you take a little bit of all the sciences, but in particular, geology and biology were the key things for me to study.
What's the most rarest fossil?
The rarest fossil? Oh, there's a lot, because there's lots of things we only have one of, right? The rarest fossil would be something we only have one example of, and there's many, many examples where we only have one. Maybe one of the most famous rare fossils, besides various human fossils, Archaeopteryx, the oldest bird, first known bird. There's only a handful of specimens of Archaeopteryx known, and what's interesting, they're all from the same site, and they're all juveniles, they're all babies. There's no adult Archaeopteryx known, and it's thought that maybe the reason for that is that the babies were learning how to fly, and some of them crashed and died in this lagoon, because they're actually preserved in a marine lagoon, and that's why we have them. And that the ones that were clever enough to learn how to fly flew away, and so we don't have fossils of them. So maybe that's probably the most famous rare fossil is Archaeopteryx, the first bird.
When the dust from the meteorite crashed to Earth and all came up, when all the plants died and all the dust settled, how would all the plants and the animals, like the mammals and the birds, all come out, and all of a sudden the plants would photosynthesize and the other animals would evolve to get used to all the dust and debris on the surface?
So how did they sort of recover?
Yes.
So how did mammals and birds and the plants recover from the asteroid impact? Well, the record we have, for starters, shows that they did recover fairly quickly, probably within a few thousand years after the impact event. We see evidence that the plant community is coming back and diversifying and reestablishing itself. So it probably took a fair amount of time, hundreds to a couple thousand years, for the plant communities to reestablish themselves. And once you have the plant community reestablished, then animals can, rediversify and fill back in again. But it probably wasn't an instantaneous process. It probably took hundreds of years for the ecosystem to reestablish, clean itself back up again, and especially for the plant communities to recover and refill in the landscape.
Like if people got extinct and then an animal had all the then a kind of animal got the technology that we do now, then would they ever then do you think they might pull up people's bodies that are fossils?
That's an interesting question. Like, will we become fossils? What is our fossil record going to look like? That's an interesting question. It's really going to we mess up our fossil record terribly, right? Because for starters, and not to be ghoulish or squeamish, but think about the cemetery, right? We're right across the street from a cemetery. People are going to look at that cemetery and they're going to see all these people, many of whom never met each other, didn't live at the same time. And anyone who comes a million years from now, animals or aliens come a million years from now, it's often aliens we talk about, aliens are going to come 3 million years from now, what are they going to see? They're going to see places like cemeteries. They're going to see people who never met each other, didn't live at the same time, all neatly lined up as if we did all live at the same time. It's called a time-averaged assemblage, that's a technical name for it, where people organisms that did not live in the same place at the same time are gathered together and artificially made to look like they did. So I think our fossil record is going to be very distorted. It's going to look very weird. We're going to have one. Of course we're going to have one. We're helping it out, actually, by burying a lot of our bodies. We're encouraging the fossilization process in that sense, if you think about it. But it's a very distorted fossil record. Is it going to be an accurate depiction of what our communities look like, about what our ecological interactions are? Where's all our food? Is our food being buried with us? If you think about a fossil record where we have the predator and the prey, we can figure out who was eating whom. Could we do that with us? All sorts of really interesting philosophical questions you could think about with that. But we'll definitely have a fossil record.
How long does it take fossils to form?
How long does it take fossils to form? It varies a lot. It can be a very long process, very slow process over thousands and thousands of years as say a bone is buried in the sediments and accumulates material, minerals filling in the pore space. On the other hand, people have actually done experiments, very, very smelly experiments, where they've taken carcasses, pig carcasses, dead shrimp, stick them in, one of my favorites, take a shrimp, don't do this, your parents will hate you. Take a shrimp, take a jar of stinky mud, like really smelly mud with lots of bacteria in it, stick the shrimp in the mud and close the jar. Go away. You come back a couple days later and you pull the shrimp out and it's going to have pyrite, fool's gold, growing on it. So within a few days you can get that pyrite coating on a fossil, a potential fossil. So it really varies quite a bit depending on the chemical conditions in the sediment. Don't do that experiment at home unless your parents say you can.
If you did make the dino chicken, would he be a meat eater? Would it turn into a meat eater?
Would it be a meat eater? Interesting question. Chickens today are really omnivores. If you let a chicken go at free range, they eat kind of whatever they encounter. They eat plant material. They also eat insects. They eat bugs and stuff. So I think it would be perfectly happy eating whatever it can catch. Which is why, children? Yeah, I think you don't want them to be that big. I'm a little nervous about the dino chicken myself. I think it would be a meat eater, or at least it would eat both potentially. It's capable digestively of eating both plant material and meat.
Don't we already have a fossil record of ourselves, like with the Mayans and cavemen?
Well, certainly we have both. We have both. We have. So I want to separate. We have a fossil record. We have the fossil record of humans, our species and other species of humans that goes back 4.5 million years. We also have an archaeological record, and I would just make a distinction. Archaeology, the study of human cultural artifacts. So the study of Mayan culture, of ancient Egyptian culture, pottery, the buildings and things like that. That's separate from paleontology, which is more the study of the bodies themselves, the biology of ancient life, as separate from the cultural artifacts. But we do absolutely have both an archaeological cultural record and a physical fossil record of humans that goes back more than 4 million years.
If there's a lot of one species, how did it knock out the entire species?
If there's a lot of
one species, how did it knock out the entire species?
The asteroid hitting the Earth? It was something that would have devastated the entire planet, and it would have definitely selectively hit certain groups of organisms more than others. So very large animals selectively died out as opposed to small animals. Animals that were dependent on photosynthesis-based food webs, things that ate plants or ate things that ate plants, died out much more than things that lived in aquatic habitats, for instance. So given the scale of the impact event, it's perfectly reasonable that it would have killed out an awful lot of things.
If the Ice Age or last ice house setting was 300 years ago, does that mean in 300 years into the future we're going to have another one, or has the climate changed too much to be able to have that happen?
Okay, so the last ice house age before ours was 300 million years ago. And then our ice house started about 2.5 million years ago. But the ice sheet expansion and contraction happens on a scale of about every 100,000 years. It goes back and forth. There is a real question right now. We've been in a time period when the ice sheet has retreated back. For about the last 10,000 years, the ice sheet has been retreating back. We're due, really, for it to start expanding again. We're due to start sliding into the next glacial expansion. But I don't think that's going to happen. Most geologists don't think we're going to have another glacial expansion. I think most geologists think that human-induced climate change is going to kick us out of the ice age altogether, that we're not going to have big ice sheets at all, or very few, and that we're not going to see this glacial expansion that we're really due for soon. The next couple thousand years, we should be going back into cold time. We're probably not going to see that because of human climate change.
Can they bring like a clone of an ankylosaurus back, like the ankylosaurus dinosaur?
Can they clone specific kinds of dinosaurs, like the ankylosaur? Yeah, specifically that one. Yeah. The challenge with cloning very old things like dinosaurs, like you saw in Jurassic Park, they needed decent DNA. You need a good sequence of DNA to do proper cloning. And you can do that with recent stuff. That's why they're trying to do it with the woolly mammoth. But with a dinosaur, there's no good DNA that's ever been extracted, at least so far, from a dinosaur of any kind, anything that old. The oldest pretty decent piece of DNA that's ever been extracted from anything, any fossil organism, there was a claim that an insect in amber that was 40 million years old, they got a pretty good piece of DNA out of that insect. But then there was some debate about whether that was really good or not. So that's about as old as anyone's ever gotten. Yeah, exactly. There is the possibility, there's a T-Rex and a duck-billed dinosaur that there's been a report that they got some protein out of a bone. Not DNA, but protein, that DNA makes, that they were able to get some sequence data out of. But it was fragmentary, wasn't a whole thing, wasn't enough to actually make a whole nother dinosaur. So maybe it's not out of the realm of possibility yet. So that's, I think, why they're doing the dino chicken, because it's a living relative of dinosaurs that still has a lot of the DNA.
I actually have two questions. One day when I was at the beach, I found a fossil in Lake Erie.
What was the first part again?
Once in Lake Erie, I found a small fossil of a shell.
Okay. Was it like the brachiopods shell? Did it look kind of like that?
It was just like a circle shell. What could you infer about that being there at that spot?
Okay. Lake Erie, the actual lake itself, formed about 3 to 5 thousand years ago. It's quite young. The lake itself is quite young. So the material in the lake could be from the Ice Age. So it could be Ice Age material. More likely, though, the bedrock, the solid rock underneath the lake is much older. And it comes from the same time, more or less the same time as the brachiopod and the trilobite I showed you. What you find very commonly in the lake, besides the brachiopod, are some fossil coral, which have a round top to them. So if you say it's round, it might be a fossil coral. That would be probably my best guess. They're really common, especially on the southern shore of the lake. I've seen lots of fossil coral, and it's pretty round looking.
Also, how could have the world gone from a greenhouse to an ice age and then back to a greenhouse?
How does that actually happen? Excellent question. Lots of debate about that. There's a lot of unanswered questions about how these major climate shifts happen. A lot of it has to do on a very long time scale with tectonic changes. I showed you Pangea breaking apart and the continents moving. Well, the continents shifting changes the whole pattern of ocean circulation, how the ocean currents are moving. Where the continents are is also affecting how much sunlight is reaching continent versus how much sunlight is reaching the ocean, because you have either ocean or continent at the equator. So there's a long-term change because of where the continents are versus where the oceans are in how the currents are moving and how sunlight, and the heat from sunlight, is being distributed around the planet. So one explanation is, on a long time scale, plate tectonics. And the shifting of the continents is actually having a long-term control on climate. Changes in volcanic activity associated with tectonics are also part of it too. A lot of volcanic activity means more carbon dioxide coming out of volcanoes, which means more warming. So, but there's a lot of other stuff going on too. It's a very complex system.
You said there was dinosaurs in the ocean, and since there's not any left, what kind of effects came off of the asteroid to kill off the aquatic dinosaurs?
Good question. So the marine reptiles, like the mosasaur and the plesiosaur, the thing that looks like the Loch Ness Monster, they also died out when the asteroid hit. And the idea there, the photosynthesis being shut down because of the darkness, because of all the debris in the atmosphere, that would have affected life on land, but it also would have affected life in the oceans, because shutting down photosynthesis shuts down all of the plankton in the ocean, which photosynthesize. And in much of the ocean, that's the basis of the ocean food web too. So things photosynthesize in the ocean, and then things eat those things, and then fish eat those things, and then the big guys eat the fish. So if the plankton shuts down, anything, especially large animals, are very vulnerable to changes in the food sources in the ocean too. That's probably what killed them off. And the ammonites. That's probably what killed off the ammonites.
All right, so we were wondering, the way you get a hybrid flower if you want a certain color is you take two other genes of two colors and mix them together. So if you want pink, then you have to mix together white and red. But sometimes it's white, and sometimes it's red, and sometimes it's pink. So with the dino chicken, wouldn't it sometimes be a chicken, and sometimes be a dinosaur, and sometimes be a dinosaur chicken?
Reasonable question. Reasonable question. I think the idea with the dino chicken is that what you're talking about is sometimes the offspring will have one gene or set of genes, and sometimes the alleles, and sometimes they'll have, so it'll be like a capital A and a capital A, and sometimes it'll be a capital A and a little a, and sometimes it'll be a little a and a little a, thinking about it that way. In this case, we're talking about a different kind of genetic pattern where during the development of, well, any of you, for instance, when you were developing as an embryo, my favorite example, you have 5 fingers, right? Right? Most of you have 5 fingers. Anybody here not have 5 fingers? I'm asking seriously. Does anybody here not have 5 fingers? Because the reason you have 5 fingers is that when you were developing as an embryo, there was a gene, and the gene's function was to turn on in the embryo and say, okay, start making fingers. And it told other genes to turn on and start making fingers in the embryo. And then that gene then said, okay, you've got enough now, turn off. And it says, okay, shut off the gene. And so it said, okay, shut off the finger-making machinery, and then it stops at 5. Well, that gene fairly frequently forgets to turn off. And so you make finger 1, 2, 3, 4, 5, 6. Oops, wait, shut off, and then it shuts off. About 1 in 500 people are born with a 6th finger. Often it's just a little stub of a finger, and the doctor just snips it off as soon as you're born. And now everyone is going to look and see if they have a scar. And maybe you should ask your parents. 1 in 500 people are born with a little stub of extra finger, because the gene that says make fingers now just stays on a little too long before it shuts off, and you start making that 6th finger. Some people have a fully formed 6th finger.
Okay.
That's what the genes are like that we're talking about.
We're going to have to stop for lunch, and I'm just going to take one more question. But the best news is that we're not allowing Dr. Pegg to escape. She'll be there in the afternoon, and you'll be able to ask her questions again.
And I'll have lots of fossils.
And we have lots of fossils. We have also some very old reburied organisms. And we have some things for people who are not as squeamish as Dr. Pegg to dissect. So last question.
One more question.
Did you know that European scientists brought back a plant from the Ice Age because a squirrel put it in his burrow and then it got frozen over and then they brought back the plant? It was really cool. My sister did a report on it.
Yes, I have heard about that. It was just reported fairly recently in the scientific literature where an Ice Age squirrel, it's totally from the movie The Ice Age. Ice Age squirrel buries a plant and they find it and now they grew it and it's growing. That's an example where something can get frozen for very long periods of time. I forget exactly how old it was. Do you know how old it was? Several tens of thousands?
Between 10 and 20,000, I think.
More than 10, less than 20,000, something like that. Years old, and they got it to grow, and it's growing. Yes, very cool.
Okay, first thing we have to do is give Dr. Pegg a really big hand. Okay, thanks so much. We'll see you about 1:15 in the other room.